Hi, I'm Dr. Molly Bindell. I teach at Thomas Jefferson University in Philadelphia, Pennsylvania. I'd like to introduce you to my students' course-embedded research project in their bioinformatics class. First, I'd like to give you a bit of background on the course specifically. It's called Applications in Molecular Biology and Bioinformatics. It's a 300-level course intended for juniors and seniors majoring in areas such as biology, biochemistry, and pre-medical studies. The course is intended to give students hands-on experience with cutting-edge tools and techniques in molecular bio, as well as experience using the latest bioinformatic tools and computer programs, all in the context of a course-long soil microbiome research project that the students themselves develop and execute. Important to note that I didn't expect any coding or bioinformatics background for this course. This course included a 3-credit lecture component and also a 1-credit co-requisite complementary lab experience. The first half of the lecture portion of the course focused on molecular biotechniques, while the latter portion of the course focused on data analysis using bioinformatic tools. I wanted to make sure that students got to design their own authentic research projects so that they could learn bioinformatics in a non-intimidating and enjoyable way that would allow them to build their confidence in bioinformatics. Allowing them to explore their own research project made it a more meaningful, fun experience for them. Students were instructed to focus their projects on the exploration of drivers of bacterial diversity in soils. They were allowed to run wild with their ideas and explore any area within that realm that interested them. They worked in small groups to find interesting to them soil microbiome studies. They then formulated their own hypothesis and designed their own soil sampling strategies. They collected soil samples, extracted DNA, ran PCR, and got their samples sequenced on a next-gen sequencing platform. They then spent the last several weeks of class learning how to analyze their big molecular datasets using R programming and a hot new student-friendly bioinformatics platform called DNA Subway. Without further ado, Wynter, Henry, and Zinnia Ruch will present their exciting work on soil microbiomes at varying soil depths in Philadelphia.
Hi, we're Zinnia Ruch and Wynter Henry, and today we'll be discussing our project, Soil Depth Impacts Bacterial Diversity in a Philadelphia Deciduous Forest. Soil microbiomes are a diverse environment of bacteria, archaea, and fungi. In this project, we look specifically at the bacterial microbiome of soil layers in the Wissahickon Valley Park, which is a forested park located in northeast Philadelphia. The main limiting factor for bacterial communities is the abundance of food sources like plant matter, so the expected microbial hotspots in this area are fallen leaf litter and the soil surrounding root systems. The Wissahickon is a deciduous forest, and there are large volumes of leaf litter on the forest ground. Therefore, we hypothesized that there would be greater bacterial diversity in the top layer of soil as compared to deeper soil regions. Here's an overview of the methods used in order to test our hypothesis from proposal to data analysis. We collected 3 soil samples above 3 centimeters, which we refer to as topsoil, and 3 samples between 28 and 30 centimeters, which we refer to as subsoil. Total genomic DNA from each soil sample was extracted using a DNeasy Power Soil Pro kit. Additionally, DNA was extracted from Bacillus, Staphylococcus, Listeria, Escherichia shigella, Enterobacteriaceae, Enterococcus, Lactobacillus to serve as a control. Then the polymerase chain reaction, also known as PCR, was performed in order to amplify the highlighted portion of the 16S rRNA gene, a common barcode gene used for identifying bacterial species. Soil samples PCR products were then sequenced on an Illumina next-gen sequencing platform, and the raw data was sent back to us. Unfortunately, one topsoil sample did not have the adequate PCR product and could not be sequenced. The 5 remaining samples data was uploaded to DNA Subway, a web-based tool that allows users and instructors to easily conduct bioinformatics techniques. In this instance, it was used for metagenomic analysis. Shannon diversity indices were calculated for each sample in order to decipher the overall diversity of each one. This was done through RStudio and R, a programming language used for statistical programming. MEGA-11 was used to generate a phylogenetic tree to place an unknown species. The results show that the topsoil had greater overall bacterial diversity compared to the subsoil samples. We found 217 species unique to only the top layer of the soil. The Shannon Diversity Index on the right, a metric for measuring species diversity in a community, is significantly higher in the topsoil samples than in the subsoil. This bar plot, representing the total taxonomic diversity present in each sample, was generated by DNA Subway, using the 16S bacterial DNA database up to a species level. The topsoil samples are the two far right columns, and the subsoil samples are the next three columns. The control group is shown on the left-hand side. The different bands within each sample's column represent relative abundance of individual taxa. The thicker the bar, the higher the relative abundance of that taxon. The topsoil samples have more bands and therefore more bacterial diversity compared to the subsoil, which has fewer but thicker bands. Overall, we determined that topsoil has greater bacterial diversity in these samples. This may be due to the presence of a wide variety of food sources and therefore less competition among species. Therefore, more species can coexist there. Additional soil sampling will allow us to provide more support to this hypothesis. However, DNA studies are limited. You can get information as to which species are active in the soil by sampling DNA. Therefore, extracting RNA from the samples would show us which bacterial species are alive and active in the soil. This can contribute more to our understanding of important ecological roles these bacteria play in our ecosystems. Thank you for listening. Have a good day.